Good evening, everyone. We'll begin in 1 minute. We'll begin in 5 minutes so that more people can join in. Thank you.
Be curious, not judgmental. I like that. So I get back in my car and I'm driving to work, and all of a sudden it hits me. All them fellas that used to belittle me, not a single one of them curious. You know, they thought they had everything all figured out, so they judged everything. They judged everyone. And I realized that they're underestimating me. who I was had nothing to do with it. Because if they were curious, they would ask questions. You know, questions like, "Have you played a lot of darts, Ted?" Which I would have answered, "Yes, sir. Every Sunday afternoon at a sports bar with my father, from age 10 till I was 16, he passed away. Barbecue sauce."
Hi there.
Yeah, I guess we can start our session today. So, as you know, we are studying about adherence molecules, cell surface molecules, which help in connecting one cell to another or one cell to the surface, right? So, we'll talk about adherence junctions, which we have talked about in the last class. It's a continuation of it only. So, adherence junctions respond to tension from inside and outside the tissue, right? Because they are connected to the intracellular filaments, correct? So it's pretty obvious, but no, it was not obvious, because if you don't know how they are connected and how they respond, we can only speculate. So there are certain experiments which takes place to assure that whatever our hypothesis is, it's actually correct. So this is actually a result. So the result is adherence junctions respond to tension from inside and outside the tissue. Okay, so the protein complexes that mediate junctions either between cells or between cells and the extracellular matrix are dynamic machines that have the remarkable ability to sense mechanical stresses and generate biochemical signals that lead to an appropriate response. We call this mechanotransduction. As we discussed in the previous sections, adherence junctions are linked through catenins to contractile bundles of actin and myosin 2. These junctions are therefore subjected to pulling forces generated by the attached actin. The pulling forces are important for junction assembly and maintenance. Inhibition of myosin activity, for example, results in the disassembly of many adherence junctions. Furthermore, the contractile forces acting on a junction in one cell are balanced by contractile forces at the junction of the neighboring cells, so that no cell pulls others towards it. Adherence junctions sense the force acting on them and modify local actin and myosin behavior to balance the forces on both sides of the junction. Evidence for these mechanisms comes from studies of pairs of cultured mammalian cells connected by adherence junctions. If contractile activity in one cell is increased experimentally, the adherence junctions linking the two cells increases in size, and the contractile activity of the second cell increases to match that of the first, resulting in a balance of forces across the junction. These and other experiments reveal that adherence junctions are not simply passive sites of protein-protein binding, but are tension sensors that regulate their behavior in response to changing mechanical conditions. Mechanotransduction at cell-cell junctions is thought to depend, at least in part, on proteins in the cadherin complex that alter their shape when stretched by tension. The protein alpha-catenin, for example, is stretched from a folded to an extended conformation when contractile activity increases at the junction. The unfolding exposes a cryptic binding site for another protein, vinculin, which promotes the recruitment of more actin to the junction. It is likely that these changes also lead to local regulation of the small GTPases that control actin and myosin behavior. By mechanisms such as this, pulling on a junction alters local actin behavior to make the junction stronger. Furthermore, as noted above, pulling on a junction in one cell will increase the contractile force generated in the attached cell. Because all the cells in an epithelium are mechanically linked through their cell-cell junctions and cytoskeletons, mechanotransduction can also work over long distances. In the developing wing of Drosophila, for example, contraction of cell in the hinge of the wing results in mechanical stresses that spread throughout the wing, triggering changes in cell movement and orientation that are important for wing formation. It is likely that tension sensor throughout the tissue detect shifts in mechanical stress and trigger a molecular response, because tension spreads quickly through all connected cells in a tissue. It provides an unusually rapid and effective signal to modify cell behavior throughout the tissue. So this is the mechanotransduction in an adherence junction. So when there is no tension in the other cell, right, then what will happen? The cadherin and the catenin will stay in the relaxed form. But as soon as the cell senses the other cell which has tension associated with it, then cell-cell junctions are able to sense increased tension and respond by strengthening their actin linkages, which you can clearly see, right? When actin filaments are pulled from within the cell by non-muscle myosin 2, the resulting force unfolds a domain in catenin, alpha catenin to be precise, thereby exposing an otherwise hidden binding site for the adapter protein vinculin. Vinculin then excuse me. Vinculin then promotes additional actin recruitment, strengthening the linkages between the junction and the cytoskeleton. Okay, I hope it is self-explanatory, correct? Now we'll talk about other types of junctions or molecules which connect cell to cell or cell to the basal membrane or to the extracellular matrix, one of which is called desmosomes. So desmosomes give epithelial mechanical strength. So what do we mean by desmosome give epithelial mechanical strength? Desmosomes are structurally similar to adherence junctions but contain specialized cadherins that link to intermediate filaments instead of actin filaments. Their main function is to provide mechanical strength. Desmosomes are important in vertebrates but are not found, for example, in Drosophila. They are present in most mature vertebrate epithelia and are particularly plentiful in tissues that are subject to high level of mechanical stress, such as heart muscle and the epidermis, the epithelium that forms the outer layer of the skin. Desmosomes typically appear as button-like spots of adhesion, riveting the cells together. Inside the cell, the bundles of rope-like intermediate filaments that are anchored to the desmosomes form a structural framework of great tensile strength with linkages to similar bundles in adjacent cells, creating a network that extends throughout the tissue. The particular type of intermediate filament attached to the desmosomes depends on the cell type. They are keratin filaments in most epithelial cells, for example, and desmin filaments in heart muscles. The importance of desmosomes is demonstrated by some forms of potentially fatal skin disease, pemphigus. Affected individuals make antibodies against one of their own desmosomal cadherin proteins. These antibodies bind to and disrupt the desmosomes that hold their epidermal cells, keratinocytes, together. This results in a severe blistering of the skin with leakage of body fluids into the loosened epithelium. So you can see here, this is the representation of desmosomes. You can say that they are connected to the intermediate filaments and via the adapter protein itself. Okay. And this is how they connect one cell to the another cell. So desmosomes connect one cell to another cell via the intermediate filaments, while hemidesmosomes connect the intermediate filaments to the basal lamina. Okay, so we'll see some brief about desmosomes. So let me share my screen with you guys. 1 minute. I hope you guys can see my screen.
So here is our epithelial tissue, and here is our connective tissue. Epithelial tissue is your outer surface. Connective tissue is your, it's the surface underneath the outer surface, underneath the epithelial cells, and then in between the epithelial tissue and connective tissue, we have our basement membrane, which is also our basal lamina. So if I were to look at it, from a zoomed out version, here is your epithelial tissues right over here, and you can see when you're looking at your epithelial tissue, do you see how there's lots of cells? Look at all the cells there. Lots and lots of cells in your epithelial tissue. Connective tissue doesn't have as many cells. It has a lot of fibers. Okay, it has a lot of fibers, and fibers are important. They keep your skin, your gums nice and tight. When you have an inflammation, the fibers actually break apart, and it starts to make your gums look very puffy. It can start to inflame your skin. For example, if you're looking at your skin, if you're looking at gums, the fibers can break apart. It can get puffy. So fibers are an important and integral structure of our tissues. Now, what I want to point out to you guys over here is two things actually. This I'm going to show you right over here. So this, let's see if I can outline it. This bump that you see over here, okay, this ridge, actually I should say, this ridge is known Rete ridges, or some people say Rete ridges. So I'm going to write that down. So this, when you look at it, and it's this part over here, okay, so the ridge that we're looking at over here is known as Rete or Rete ridges. And then when you're looking at it, so this is when you're looking at it from this view down, when you're looking at it going up, okay, so when you're looking at it going up, so if I'm looking at this area over here and I'm looking at it from up, that is known as your connective, is that known as a papilla? So it's actually known as your connective tissue papillae, or papillae. So all these over here, this bump that you see that's going up, that bump going up is your papilla or connective tissue papillae, and the bump going down, so this bump over here, is your Rete or Rete ridges. Okay, another thing to point out over here is, as we said, this is your epithelial tissue, this is your connective tissue, and if you guys remember, there is a layer that is separating, the epithelial tissues from your connective tissue. Do you guys remember what that layer is? Yeah, that is your basal lamina or basement membrane. So there is a layer separating between the two, between the epithelial tissue and connective tissue, known as your basal lamina. All right, good. Another thing to note is that connective tissue is also known as lamina propria. So if you hear the word lamina propria, all it's referring to is that layer underneath your epithelial tissue, your connective tissue, where you have minimal cells. You have more cells here in the epithelial tissue, so minimal cells here, lots of fibers, and there's also blood supply in your connective tissue area over here. One more thing I want to talk about is something called cell junction. So there's something called desmosomes and hemidesmosomes. So I'm going to look at it by using this visual over here. So when we're looking at this picture over here, okay, this is where when you have two cells that are connected by something, and that something is called a desmosome. So I'm going to write this down over here. Desmosome. So this over here, the cells are connected by a desmosome. So two neighboring epithelial cells are connected together by a desmosome. And here we see another desmosome, here we see another desmosome, here we see another desmosome. So basically it is cell-to-cell connection, and that's what we're seeing here. Two cells are being connected, I'm just going to write this down, by a desmosome. So, and that's kind of what it looks like. It kind of snaps together. Okay, like that. It's kind of like I've read how, how if you have a denim jacket and you're buttoning up the denim jacket, it snaps together. And that's exactly what's happening here. It's snapping together via a desmosome. Now, if you look over here, and you can see this is half of a desmosome, right? Because if you look at a desmosome, if I were to draw this out, a desmosome looks like this. Okay? So we have like two sections. Okay? A hemidesmosome looks like this. It's half of a desmosome. And so when we're looking at a hemi stands for half. When we're looking at a half a desmosome or a hemidesmosome, I'm actually going to write this down over here so that we can know how it's spelled out. So a hemidesmosome. Okay, there we go. Hemidesmosome. It's a half a desmosome. Now, what you need to know about a hemidesmosome is that it connects the cell to the basal lamina. So when the cell is connected to the basal lamina, that is known as a hemidesmosome. Okay, so this is a hemidesmosome, and this is a cell to basal lamina connection. Basal lamina connection. Separate these because it gets messy. So when you have a cell to basal lamina connection, and the cell is connecting to the basal lamina, here a cell to basal lamina, here's another hemidesmosome. Cells connecting to the basal lamina, here's another hemidesmosome. It's half a desmosome, right? That is considered a hemidesmosome. So the reason why the cells are nicely packed together over here is because of the desmosome connection. And then the reason why the cell is nicely connected to the connective tissue is because of the hemidesmosomes. When the cell is connected to the basal lamina or basement membrane, which is then connected to the connective tissue.
Hi, I took more than 300 interviews when I worked for Facebook. 80% of people I interviewed for problem solving failed the interview. 90% of people I interviewed for
Yeah, so you got that basic idea, right? That hemidesmosomes will connect the cell to the surface, while desmosomes
So in this video,
will,
I want to talk about the difference.
while desmosomes will connect the cell to the adjacent cell. So we might not dig more in this. So as till now, so we have talked about adherence junctions, we have talked about desmosomes and somewhat hemidesmosomes also. Now another type of junction which is present between adjacent cells is called tight junction. So tight junctions forms a seal between cells and a fence between plasma membrane domain. So what do we mean by this? So sheets of epithelial cells enclose and partition the animal body, lining all its surfaces and cavities, and creating internal compartments where specialized process occurs. The epithelial sheet seems to be one of the inventions that lie at the origin of animal evolution, diversifying in a huge variety of ways but retaining an organization that is based on a set of conserved molecular mechanisms. Especially all epithelial cells are anchored to other tissue on one side, the basal side. So we'll see the diagram in the coming slide. Okay, and free of such attachment on top side or the other side, the opposite side, which is facing the lumen most likely. So a basal lamina lies at the interface with the underlying tissue, mediating the attachment, while the apical surface of the epithelium is generally bathed in extracellular fluid. Thus, as discussed earlier, I forgot to change that, all epithelial are structurally polarized, and polarized means they have a basal side and they have a apical side, so they are polar in nature, and so are their individual cells. The basal end of a cell adherent to the basal lamina below, and the apical end which is exposed to the extracellular medium. Okay, correspondingly all epithelia have at least one function in common. So epithelia means normal cells, which we are talking about here is the one which is attached to the surface at the basal lamina, and one side is free, which is called the apical side. So they serve as selective permeability barrier, separating the fluid that permeates the tissue on their basal side from fluid with a different chemical composition on their apical side. This barrier function requires that the adjacent cells be sealed together by tight junctions so that molecules cannot leak freely across the cell sheet. The epithelium of the small intestine provides a good illustration of tight junction, structure, and function. This epithelium has a simple columnar structure, that is, it consists of a single layer of tall elongated cells. These are of several differentiated types, but the majority are absorptive cells, specialized for uptake of nutrients from the internal cavity or lumen or the gut. So before going into details, this is the basic overview of these apical cells or epithelial cell, which have a polarity, that is a side which is attached to the surface, attached to the basal lamina, and a free surface which is exposed to the lumen or to the extracellular fluid. So the role of the tight junction is in transcellular transport. So here, for clarity, only the tight junctions are shown. So transport, these are the GLUT transporters, right? And these are the tight junctions you can see. So this is one cell, this is another cell, and this is third cell. All these three cells are connected to each other via tight junctions. So the transport proteins are confined to different regions of the plasma membrane in epithelial cells of the small intestine. This segregation results in the one-way transfer of nutrients across the epithelium from the gut lumen to the blood. In the example shown, glucose is actively transported into the cell by sodium-driven glucose transporters at its apical surface, and it leaves the cell through passive glucose transporters in its basolateral membrane. Tight junctions are thought to confine the transport proteins to their appropriate membrane domains by acting as diffusion barriers or fences within the lipid bilayer of the plasma membrane. These junctions also block the backflow of glucose from the basal side of the epithelium into the gut lumen. Okay, so we'll just go to the details of this. So I hope you understood this. What type of cells we are talking about? We are talking about cells which have polarity, and by polarity I mean that these cells once they will on one side they will be connected to a rigid basal lamina, and the other side will be free for them to move. And generally these cells, if they are facing the lumen or they are in the intestine, they'll help in transporting of nutrients. So one example we saw here is the transportation of glucose from the lumen to the intestinal cells, and finally these intestinal cells will send these nutrients to the blood, and then it will go to different cells. So the absorptive cells have to transport selected nutrients across the epithelium from the lumen into the extracellular fluid on the other side. From there, these nutrients diffuse into small blood vessels to provide nourishment to the organism. This transcellular transport depends on two sets of transport proteins in the plasma membrane of the absorptive cells. One set is confined to the apical surface of the cells, that is facing the lumen, and selected molecules into the cells from the gut, that is the lumen space. The other set is confined to the basolateral or basal and lateral surface of the cell, and it allows the same molecules to leave the cell by passive transport into the extracellular fluid on the other side of the epithelium. For this transport activity to be effective, the spaces between the epithelial cells must be tightly sealed so that the transported molecules cannot leak back into the gut lumen through these spaces. Moreover, the transport proteins must be correctly distributed in the plasma membrane. The apical transporters must be delivered to the apical membrane and must not be allowed to drift to the basolateral membrane. I guess we can take this topic in the next class. So we'll try to summarize the type of CAMs we have studied till now, their basic role, and then we'll get into little bit of more detail for the tight junctions. And then most likely we'll end our discussion on hemidesmosomes or other type of like gap junctions are already also there. So I'll see the extent of information which should be there for you guys to have a little bit idea of all the connections which these cells form and the proteins which are associated in making these connections. Okay, so for today I guess I'll stop my class here itself. So today was not such an elaborate class, but I hope that you guys are keeping up with the pace, and if you have any queries, any doubts, feel free to drop me a mail. Thank you so much.